All right, folks, welcome back to the respiratory system. This mini lecture is still quite a bit of anatomy in it. We're going to skip through pretty quickly. You've already had the anatomy, but it does start with some of the physiology. So I'll skip through most of this anatomy relatively quickly and then slow down on the physiology. This is probably worth talking about. The pleural membrane surrounding the lungs. It's like a fist in a balloon, or a lung in a balloon in this case. And you'll recall that with the serous membranes, we have a visceral membrane directly touching the organ, and we have a parietal membrane that's not touching the organ. In the case of the lungs, the visceral and parietal membranes are called pleural membranes, so visceral and parietal pleura. And this space in here is the pleural cavity, but it's a potential space, not a real space. It has a thin, watery fluid in it, and it's a serous fluid. And we do have to talk about this space when we talk about pressure changes, because the pressure is transduced across this space. And of course, if something does get into this space, like air or blood, your lung collapses. You have a hemothorax or a pneumothorax. Just anatomy showing you the lobes of the lungs. More anatomy. Of course, the right lung has 3 lobes and the left lung has 2 lobes. That's the collage. It's a different section. This is a horizontal section or a cross section showing you the lungs and how the heart sits in the mediastinum. More anatomy here. Okay. What we have here, if you go back and look at these bronchopulmonary segments, what happens is you'll have a bronchopulmonary segment made by these terminal bronchioles, the respiratory bronchioles, and all the alveoli. That's a bronchopulmonary segment. All right. Different lobes of the lung have different numbers of bronchopulmonary segments. It is important clinically that we isolate lesions of the lungs so we can talk about which pulmonary segment the lesion is in. So it's not so much important for our class other than just realize that, yeah, these bronchi become, primary bronchi become secondary and tertiary and bronchioles, and eventually we have a bronchopulmonary segments. So we got these bronchopulmonary segments here. Notice the cartilage is there actually is some cartilage here, just so you know. It's just not showing it. But the cartilage is becoming more scarce, and smooth muscles are starting to surround these terminal bronchioles and respiratory bronchioles. And this is what this is showing you. This is saying, okay, here's my bronchopulmonary segment right here. That's what it's saying. And what is that? Well, it's the smaller bronchioles. Actually, it's saying the smaller bronchi, then the bronchioles, terminal bronchioles, and respiratory bronchioles. And you can notice that with the smaller bronchi, the cartilage becomes less and less and less. These blue or turquoise colored things are cartilage. And what I do is I get to increase the smooth muscle. And ultimately, I end in an alveolar sac. And this is where most of the gas exchange occurs. It is true that some gas exchange can occur in the respiratory bronchiole, but most of it occurs here in the alveolar sac. So that's a bronchopulmonary segment. This is showing you a couple different things. First of all, here's my pulmonary artery bringing blood to my lungs. All right, and it keeps bifurcating until it becomes a capillary bed. And then here's the pulmonary vein taking the blood back to my heart. Notice the pulmonary artery is blue and the pulmonary vein is red. That goes against the common rule, because from the right side of our heart, we pump deoxygenated blood to the lungs to pick up oxygen. So the pulmonary artery is blue. It hasn't picked up oxygen yet. But the pulmonary artery, the pulmonary vein is red because it's already picked up oxygen. So this is where gas exchange is occurring, right here where the capillaries surround the alveoli. Notice the smooth muscle and elastic fibers surrounding the smooth muscles surrounding the respiratory bronchioles, the elastic fibers surrounding the alveoli. So when you breathe and these alveoli expand, there are elastic fibers that recoil to bring the alveoli back to normal shape. See how the visceral pleura is touching the alveoli? And then you have the parietal pleura, which is lining the inside of your chest cavity, and here's this potential space in between it. All right, so this is pretty important anatomy right here because what we're going to talk about is gas exchange, and it's happening right here in the alveoli. That's just a blown up picture of it. That's a micrograph of it, an electron micrograph of it. Okay, so we're going to start talking about gas exchange in the alveolus here shortly. Notice the capillary bed surrounding the alveoli. That's the blood that is going to pick up oxygen and release CO2. All right, here we go. Let's start talking about the alveoli. Three major cells in the alveoli. There's type 1 pneumocytes. These are just squamous epithelial cells. They are the cells that line the alveoli. So type 1 pneumocyte here, another one here, another one here, here. Skip that. That's an alveol. That's a type 2 pneumocyte. Here, here, here. See how these type 1 pneumocytes are just lining the alveolar sacs? So this is a type 1 pneumocyte. This is a type 1 pneumocyte. This is a type 1 pneumocyte. This is. So the type 1 pneumocytes have the greatest surface area in the alveoli, and they line the alveoli. And then I have these type 2 pneumocytes. These are intercalated within the type 1 pneumocytes, and these secrete surfactant. I'll tell you what surfactant is in a second. Another type 2 pneumocyte surfactant. So type 2 pneumocytes secrete surfactant, very important. Type 1 pneumocytes are simple squamous epithelium that line the alveoli. And lastly, I have these mobile cells called alveolar macrophages. This one's in the process of moving. And these just crawl around the lungs, phagocytosizing debris and keeping your lungs clean, and they fight infection. All right, so what's surfactant? Surfactant is a detergent, almost like soap, that decreases the surface tension in your alveoli. All right, what's surface tension? Surface tension is why you could float a needle on water, why some insects can walk on water. It's why when you have two microscope slides stuck together with a thin film of water, it's hard to pull apart. That's surface tension. So when you exhale and these alveoli collapse, because remember there's elastic fibers surrounding them that recoil, there's moisture in here. And with high surface tension, it would be very difficult to take in another breath of air because the alveoli would stick to itself collapsed. But we secrete this surfactant, which is like a detergent or a soap, and that decreases the surface tension of the moisture. So now my next breath is easier. And in fact, the lungs are one of the last organs to fully mature in a fetus, and the surfactant isn't secreted in a significant amount until right at 9 months. So it's actually kind of a little race going on between can the baby produce enough surfactant to take its first breath after it's born. And this is why with premature births, the doctors and nurses usually have to intervene to help the baby breathe because the baby's lungs haven't produced enough surfactant. So that's one of the last things that's produced right before birth. Oh, and by the way, there's some research that suggests that the epinephrine secreted by the baby's adrenal glands during the stress of childbirth is needed to have your surge of for the baby to have its surge of surfactant secreted. So with C-sections, the baby might not have the epinephrine secreted to lead to the surfactant secretion. So there might be an issue there. because it seems there's suggestions that the epinephrine from childbirth, the stress of childbirth, is stressful for the mom and the baby, is needed. So memorize those three types of cells: type 1 pneumocytes, type 2 pneumocytes, and the alveolar macrophages. By the way, the alveolar macrophages are the most abundant cells in the lungs. They're the most abundant. They don't have the most surface area like type 1 pneumocytes do, but they are the most abundant cells in the lungs. So there's lots of them floating around. Here is the respiratory membrane. The air has to cross this membrane to get into the blood. All right, so what does the membrane look like? Well, first of all, you have this. This is your surfactant right here. That's your surfactant. And you have your cells. These are your type 1 pneumocytes. You have the basement membrane of your type 1 pneumocyte. And then you have the endothelial cells of your blood vessel and the basement membrane of the endothelial cells of your blood vessel. Now you're going to say it looks like one basement membrane. I'm saying there's two. These are really two fused basement membranes. It's the basement membrane of the endothelial cells, and it's the basement membrane of the type 1 pneumocytes. So it's really fused basement membrane, so you can think of it as one basement membrane. Then we have our endothelial cells. Then we have our plasma, then a red blood cell. So CO2 has to be able to traverse this, and oxygen has to be able to traverse this. The whole thing from here to here is only about 0.5 micrometers. So that's there's 1,000 micrometers in a millimeter, and this is only half of one of them. So it is very, very thin. So it's not hard for the gas to cross through here. But any kind of lesions that thicken this would decrease the gas exchange potential. All right, so that's the respiratory membrane. This is a collage showing you a zoomed in picture of the respiratory membrane. See how it's taking this box right here and it's sending it over here and saying that's a zoomed in picture. So that shows you the respiratory membrane. All right. That's it. When we come back, we're going to be talking about what causes these gases to move across that membrane.